Welcome back. This is day 3 of genetics, and today we're going to be talking about how does DNA transcribe its information outside the nucleus. So when you see this word transcribe, this is just a fancy way of saying write something down or copy it. So you may have heard about people called scribes in your history class. These were people who were in charge of writing down or copying really important historical documents to make sure they weren't lost. And the same thing happens inside your cells, except this important historical document that's being written down or copied in your body is known as DNA. So this is the beginning of a process known as protein synthesis. And if we break down that word, we're just talking about the creation of proteins. And we're going to break that process down into 2 different steps. The first step is known as transcription. And the second step is known as translation. But today we're going to be focusing on step 1, transcription, right? How this DNA is written down or copied. But I'm getting ahead of myself. Before we get into that, let's go ahead and pause this video. I want you to complete the complementary DNA strands to the following template strands. So remember your base pair rules. Go ahead and pause this video, answer questions 1 and 2, and press play when you're ready to go over it. All right. If you followed those base pair rules, remember A pairs up with T and G pairs up with C. You should have gotten the following strand, TTC, AGA, CGT. Same thing for strand B, or I'm sorry, strand number 2. You should have gotten GCA, TCA, GTA. Now, notice that these strands are separated in bunches or groups of 3. Remember, when we have a series of 3 nucleotides, that is known as a codon. So this is a series of 3 nucleotides that codes for a specific amino acid. And aren't amino acids the building blocks of proteins? Something to ponder. But before we get into that, I want us to review just some of the basic structure of DNA and proteins and what we need to know in order to understand transcription. So we know that DNA has the information to build proteins, right? Specifically, our genes contain this information. Those genes contain the codes to code for specific traits or specific physical characteristics that you will have. So here's a strand of DNA. We know that those genes are going to code for specific proteins. Proteins build up your cells, and then cells build up your entire body. So although we like to think of DNA as this magical answer, we don't really give proteins enough credit for all the work that they do. So DNA does have the instructions, but proteins are doing all of the work and are literally physically building up your body, and they deserve some credit for that. But how do proteins do all the work? Well, there's 2 separate things we need to talk about. So proteins are in charge of running living organisms, specifically through 1, enzymes, right? Enzymes are proteins. And these enzymes control all of the chemical reactions inside your living body right now. Okay, so things like digestion are controlled by enzymes. They help you function. And we also need to give proteins credit for structure. Your body is made up of several layers of very complicated proteins. So all living organisms are built out of proteins. So proteins are involved with both controlling chemical reactions as well as literally building up your body and providing you structure. Now, let's review cell organization. So we know that DNA is housed within one specific organelle inside your cells, and that is the nucleus. Okay, so let's review where that is. Here's our cell. Our cytoplasm is this blue section, but then we've got a separate yellow section right here known as the nucleus. So the nucleus is actually separated from the rest of the cell. There's a physical barrier around it. And the reason that there's a barrier surrounding that nucleus is because we want to protect the DNA that's inside. DNA is made up of genes that give us the instructions for making proteins, which is a super, super important job. So it's super important that we keep the DNA inside that nucleus where it's protected. It's basically locked away in a vault or a safe to make sure it stays safe. But then remember, what do we know about proteins? Well, proteins are made up of amino acids. There's a chain of amino acids. And those amino acids are then going to code for or come together to form proteins. But proteins are actually made by something known as a protein factory in the cytoplasm. Think about, what is that organelle called that makes proteins? I think it starts with an R. It's a ribosome. So we know that proteins are made in the ribosomes, in the cytoplasm. But we also know that those proteins get their instructions from DNA. So how do we get this DNA that's locked away inside the vault out into the cytoplasm to go to the ribosomes if we want to keep the DNA protected? That DNA can't leave, so what has to happen, Well, let's find out. So that DNA actually has to get some help because it can't leave. It's basically like Rapunzel locked away in a tower. That DNA is not allowed to leave the nucleus. So it has to call a friend. And that friend is known as messenger RNA or mRNA. We've actually heard about this molecule when we talked about mRNA vaccines. So this is messenger RNA. It is essentially a copy of DNA. So we need to get this DNA information from the nucleus to the cytoplasm, but again, DNA can't leave. So this is where we make a copy, we transcribe or write down that information from DNA onto a copy of messenger mRNA. So what happens is this blueprint of DNA stays inside the nucleus, but the mRNA is like, I got you. I'll make a copy. I'll take it over to the ribosomes and I'll tell them what to do. We're good. I got you. I'll be the messenger here. So that messenger actually carries that information over to the ribosomes. So DNA doesn't actually have to leave the nucleus. And this is the process known as transcription. We're using DNA to make mRNA, messenger RNA, and this is a copy of DNA. Right? So the DNA strand is used as a template to make mRNA. But I've got one important thing to tell you guys. Remember we talked about our base pair rules of DNA, right? We said that with DNA, A always pairs with T and G always pairs with C. But I've got one tiny exception for you, and I'm sorry to tell you that our mRNA, as much as we love it, it doesn't actually have the base thymine. So whenever you see an mRNA strand, G is still going to pair with C. Let me write that a little bit further down. But instead of A pairing with T, we're actually going to see A pair with U in mRNA. But I've got a way to help you guys remember this. Don't panic. So there's an M in front of this mRNA strand, right? Whenever you see this mRNA, you can think of Miss Collins, or even my first name is Madison, okay? So both of those are M's for you to remember. And I attended the lovely university called Auburn University. And conveniently, that's an A and a U. So whenever you see this M for mRNA, think of me, and think of me attending Auburn University, so M-A-U. Okay. Whenever you see DNA, it's still going to be the T, but whenever you see RNA, we're replacing that T with a U, which is known as uracil. This molecule of uracil is only found in RNA molecules. There is no T. So U is always going to match with A instead of T, just M in mRNA. Right? So yesterday or last week, we talked about the process known as DNA replication. We took DNA and we used it to make another copy of DNA. And we also talked about an enzyme. Go back. Oh my word, what did I do? I broke it. Okay, I'm not moving. So last week we talked about DNA replication. Right? DNA is used to make more DNA. And we also talked about an enzyme known as DNA polymerase. that was involved with this process. Remember, DNA polymerase is really good at unzipping DNA and matching bases. So it knew that A pairs with T and C pairs with G. Now, whenever we're talking about transcription, we're talking about DNA being used to make a copy of mRNA. And we still have a polymerase involved with this process, except this time, it's going to be known as RNA polymerase. But because it's a polymerase, it still has the same job. It's still going to unzip DNA and match bases. So let's figure out how that happens. But before we get to that, let's look at what the differences between RNA and DNA structures are. So DNA stands for deoxyribonucleic acid. You might see that this is a double-stranded molecule. But RNA stands for ribonucleic acid, and you may see that this is just a single-stranded molecule. And there's a reason for that. So RNA can only be one strand because it has to be small enough to fit outside of the nucleus, or really to squeeze outside of the nucleus. All right, so it has to be pretty small to fit through those nuclear pores, or like the little holes inside the nucleus wall. Okay, but other than that, DNA and RNA are pretty similar. They both use cytosine, they both use guanine, they both use adenine. The only other difference, as we just talked about, is that RNA uses uracil while DNA uses thymine. So when we're talking about base pairs with DNA, we've got G, C, A, and T. When we talk about them with RNA, we've got C, G, A, and U. All right, so the only other difference there is that when DNA uses a T, RNA is going to use a U. All right. But now let's look at how this actually happens. So how do we match the bases of DNA and RNA? Well, the first thing that has to happen is that double-stranded DNA has to unzip. And it has to unzip so that enzyme RNA polymerase can get inside. Right? And RNA polymerase is going to run along this base sequence of DNA and do what it does best, match those RNA bases to DNA based on those base pair rules. So it's going to run along that DNA strand and see, okay, all right, I see a C, that means I need a G. I see a T, that means I need an A. And it's going to synthesize or basically copy that genetic information using those base pairing rules. So A with U, G with C. And what happens is it will actually synthesize one single stranded copy of that DNA sequence. So I want you guys to actually pause this video. I know this example is not on your paper, but I want to challenge you. I want you to see if you can copy this sequence of mRNA as if you were RNA polymerase. So if you saw this DNA strand and you were mRNA polymerase, what would you copy down? All right, pause this video, make that matching sequence, and press play when you're ready to review it. All right, so remember that instead of T, U is matched to A instead. So A is always going to match with U, G is going to match with C. So that DNA strand is going to get copied by RNA polymerase, and that mRNA sequence is going to be as follows. Ooh, so slick. So notice that the only difference here is that we use U's instead of T's. Everything else is matched the same. So whenever we see a T, we're still going to match up an A. Whenever we see an A, the only difference is we add a U. Okay, so what happens now? Well, we've made a copy of the DNA, but what does that mRNA need to do now? Well, it's got the message, it's got the important information. Where does it take it to? Well, it has to go to the ribosomes. So what ends up happening is that strand of mRNA is carried over to the ribosomes. And that it's actually kind of a dramatic process. Okay, imagine like the mRNA is like sprinting. Okay, it's sprinting through the cell. It's like, I've got to get this information over to the ribosomes. We've got to build that protein. And it gets this. By the time it gets to the ribosome, it's fully out of breath. It's like, here, take this ribosome. I need you to build this protein. And the ribosome's like, all right, let me get my glasses. Got it. let's see what we're making today. And it reads that mRNA sequence. And then what happens? Well, that ribosome does what it does best, and that is it's going to synthesize a chain of amino acids according to the codons that were present. So remember, we talked about codons. These are sequences of 3 nucleotides that call for a specific amino acid. So the ribosome is going to put all of these amino acids together, and when you have a long chain of amino acids, we're overall synthesizing, what's that molecule called? A protein. All right, I feel insane sitting here talking to myself. So let's go through this process one more time, because I know it can be super confusing. So to review, we've got the DNA, super important molecule, locked away inside the nucleus. It's got the blueprint for how to build proteins. But we build proteins in the cytoplasm, in the ribosomes, but that DNA can't leave. So it has to get the help of this mRNA copy. So mRNA copies that important DNA molecule. And then it carries that copy over to the ribosomes, which are going to read that strand of mRNA and then churn out the amino acids that are coded for by those codons. Once those amino acids are built together into this long chain, we're going to form a protein, which of course is going to code for the traits that you currently possess. So this process right here, where we go from DNA to mRNA, is called transcription. A copy is written down. Tomorrow, we're going to be talking more about this second part of the process right here, where we go from mRNA to protein. This is the process that's known as translation. Again, we'll talk more about that tomorrow. So this entire process as a whole is known as, you guessed it, protein synthesis. All right. And I think that's all I have for you guys today. Yes, so there is, wait for it. I'm really struggling with these PowerPoints today. Okay. Come on, we're almost there. I just have one more thing to tell you guys. All right, there is a written passage at the end of your worksheet today. Don't worry about it. We're gonna go over this in class tomorrow. So please do not worry about reading that passage or answering the questions. Again, we will do this together in class. So the only other thing you need to do is fill out your exit ticket, submit these notes and your exit ticket, and let me know if you have any questions. So transcription and translation can be pretty confusing. So we're gonna spend the next few days breaking it down and really making sense of this whole process. But other than that, have a wonderful day, and I will see you guys tomorrow.